Swayam Prabha. Digital India. Educated India.
Namaste. Today we move with the second part of the revision. So, we begin with the fourth module, which was ecological energetics. It had 3 lectures: food chains, food webs and trophic levels, primary production and nutrient cycles. So, in the case of food chains, this is one image that we know from our school days. So, grass is eaten by insects, insect is eaten by the frog, frog is eaten by the snake, snake is eaten by the eagle, and the grass is making food by using the sun's energy for photosynthesis. So, this is an example of a food chain. It is defined as the transfer of food energy from its source in plants through herbivores to carnivores, detritivores or decomposers, and this is referred to as a food chain. Then we defined autotroph and heterotroph. Autotroph is an organism that can make its own food. Heterotroph is an organism that needs to eat some other organism for its food, or to eat some other part of an organism for its food, because it cannot make its own food. Autotrophs are divided into photoautotrophs and chemoautotrophs, depending on whether they are using light for making their food, or they are using some chemical reactions for making their food. Then we define producers and consumers. Producer is an organism that makes its own food. Consumer is an organism that consumes some other organism for food. Then we have consumers are divided into primary consumer, secondary consumer, tertiary, quaternary and so on. So, primary is the one that is feeding on the producers, secondary feeds on primary, tertiary feeds on the secondary, quaternary feeds on the tertiary and so on. Next we defined herbivore and carnivore. So, herbivore is an organism that eats only plants, so they are the primary consumers, and carnivore is an organism that eats other animals, so they can be secondary consumers, tertiary consumers or quaternary consumers such as tigers. Then we have omnivore that eats both plants and animals such as bear, and decomposer is an organism that converts dead material into soil and recycles the nutrients. So, it plays a very important part in the functioning of the ecosystem, and it includes detritivores and microorganisms. Detritivores are those organisms that feed on detritus, which is decomposing plant and animal parts as well as feces, and make it more exposed to the action of microbial decomposers such as bacteria and fungi that further break it down. Then we looked at these 2 kinds of food chains. One is the grazing food chain, which starts from a plant base, and detritus food chain that starts from a detritus base. Now, grazing food chains have 2 categories. We have the predator food chains, in which the size of the organisms increases as you move up the food chain, and the second one is a parasitic food chain or a parasite food chain, in which the size of the organism reduces as we move up. Then we had a look at the differences, and typically detritus food chains are shorter chains because they do not have sufficient energy to support a very long chain. Then we looked at food webs. So, food web is a system of interlocking and interdependent food chains, and each of the several hierarchical levels in the ecosystem is known as a trophic level. So, in this case this is a food web, and in this food web you have these organisms that are eating different organisms, and in turn are getting eaten by different organisms, and in actuality a number of food webs are extremely complicated. Next we had a look at ecological pyramids, which is a graphical representation that is designed to show the biomass, number or energy at each trophic level in a given ecosystem, and it is also known as a trophic pyramid or an Eltonian pyramid or an energy pyramid or a food pyramid. And then we looked at pyramid of numbers, how many organisms are there at each trophic level. It can be inverted, in which case you have one plant that is feeding a number of herbivores, which is feeding an even larger number of carnivores and so on. Or you can have pyramid of numbers. So, this is an example where you have a tree, and this one tree is feeding a number of birds. Each of these birds has different parasites, and each of these parasites have a number of hyperparasites. So, in this case the pyramid of numbers is inverted. Then you can have a spindle pyramid of numbers, in which case you have a tree with a number of frugivorous birds which are being eaten upon by a hawk. or you can have some other spindle pyramid of numbers like like phytoplankton, zooplankton, fish and sea lion. Here again it is spindle in shape, because the number of sea lions is less than the number of fish, is less than the number of zooplanktons, but in this case you have a small number of phytoplanktons that are supporting a larger number of zooplanktons, probably because the phytoplanktons are reproducing at a much faster rate as compared to the zooplanktons. It can even be a dumbbell shape, such as grass, rabbit and fleas. The second one is a pyramid of energy, which measures the energy that is contained in organisms at each trophic level, and this is typically an upright pyramid. And then third is the pyramid of biomass, which is the biomass of all the organisms in each trophic level. It can be upright or it can be inverted, as in the case of an oceanic pyramid, in which case you have planktons which have a smaller biomass as compared to the small fishes, then large fishes and then even larger fishes, or maybe some larger mammals, such as if you have a blue whale, so the biomass will be very high. Then we defined standing crop as the total dried biomass of the living organisms that is present at a trophic level, and we also defined ecological efficiency as the efficiency with which energy is transferred from one trophic level to the next. We defined exploitation efficiency, assimilation efficiency, gross production efficiency, net production efficiency, and then we defined ecological efficiency as a product of three of these. Then we had to look at the 10% rule. So, whenever energy is being transferred from one trophic level to the next trophic level, typically only 10% of the energy is stored in the next trophic level, and as much as 90% is lost during transfer due to incomplete digestion or as part of the respiration. So, typically if you have a tiger that is chasing a sambar, so the tiger is using up energy during this process because it is expending energy in chasing the sambar. Then the tiger will not be able to eat all the body parts of the sambar. There will be some amount of incomplete digestion, and plus the tiger is also respiring, it is also using energy for its own biological processes, so it will also lose out some other parts of energy. So, typically 10% is passed, except in the case of the very first stage where the plants have typically 1% of efficiency. So, if you have 100,000 joules from the sun, only 1000 joules, that is 1% of that energy, is stored in the grass. So, we can also write the 10% rule in this equation. Then we looked at trophic cascade, an ecological phenomenon that is triggered by addition or removal of top predators and involving reciprocal changes in the relative populations of predator and prey through a food chain, which often results in dramatic changes in ecosystem structure and the nutrient cycling. So, in this case, if you make changes in the top of the food chain or the top of a food web, then these changes get trickled down along the whole of the food web. And a good example is the wolf case study that we saw, in which case when wolves come into the national park, they are able to change the behavior of the deers, because of which deers move away, and because of which a number of plants are able to survive and they are able to grow, because of which you get more amount of berries. So, you start getting bears in that area, you start getting birds in that area, and so on. So, just the addition of wolves makes a huge difference in the whole of the food web. Then next we looked at primary production, which is the synthesis of organic compounds from atmospheric or aqueous carbon dioxide through the process of photosynthesis or chemosynthesis. We have defined photoautotrophs, and the importance is that it is releasing oxygen as a byproduct, it is supporting the whole of the ecosystem, and plants form a very large part of the earth's living mantle. So, we looked at these two processes, photosynthesis and respiration, which are essentially inverse of each other, and then we defined gross primary production, net primary production, and compensation point. So, compensation point is the equilibrium point for plants where photosynthesis is equal to respiration, and typically this occurs in early mornings and late evenings, where the plants are doing the photosynthesis, but at the same time the photosynthesis is not so large that it would be able to counter the impacts of respiration. During the daytime, typically you will have situations where photosynthesis is greater than respiration. In the night time, respiration is greater than photosynthesis. Then we can measure the the production by a number of ways. We can look at the amount of gases that are utilized, the amount of gases that are released, or we can look at the amount of radioactive materials that get incorporated by labeling the carbon dioxide, or we can look at the harvest or the total amount that is stored in the plants in a particular time frame. Then we defined efficiency. You have you have gross efficiency and you have the net efficiency. So, in the case of the net efficiency, you deduct the amount of energy that is fixed, which is now released because of respiration. Then productivity is defined as production per unit time, and we define net primary productivity as APAR into LUE. APAR is absorbed photosynthetically active radiation, LUE is light use efficiency, and by using these we can make use of satellite data or modeling data to look at these different values. And productivity depends on a number of factors. It depends on the species that you have, it depends on the amount of sunlight that you are getting, which will again depend on the latitude and the cloudiness of that area. It will depend on the dust and water that is there in the environment, the fertility of that area, the leaf arrangement, the leaf area, the concentration of carbon dioxide, and so on, and a number of other variables. And then we looked at this use of modeling, through which we can set up a model by which we are able to match the field characteristics that are observed in different plants. And once you are able to model all these characteristics, then you can ask different questions like what are the impacts of global warming on these different plants. So, when you have global warming, there is an increase in the concentration of carbon dioxide in the atmosphere, which will lead to an impact that is known as carbon dioxide fertilization, because you have more carbon dioxide. So, you will have more amount of productivity. On the other hand, because of changes in climate, you can have a positive impact or a negative impact. So, for every species, for every location on this planet, you can compute whether this global warming is going to harm your plant species or whether it is going to benefit the plant species. And we can also see how the timing of harvest changes for a number of organisms. So, in this case we see that in the current situations, if the harvest period is say 110 years, after you have this global warming, it may shift to as low as 75 years. Next, we defined oligotrophic lakes, mesotrophic lakes, and eutrophic lakes. So, oligotrophic lake is that has low nutrient content, mesotrophic lake has an intermediate level of productivity, intermediate amount of nutrients, and eutrophic lake that has a high amount of productivity, high amount of nutrients, and hypereutrophic lake that has even more. Then we looked at different characteristics, especially the Secchi depth. So, Secchi depth is a measure of the amount of turbidity that is there in water, and if you have a hypereutrophic lake, the turbidity will be so high that the Secchi depth will be very less. Then we looked at the sources of nutrients: dust, bird droppings, upwelling, rivers bringing sediments, and so on. And then there is this role of the limiting nutrients. So, if there is a nutrient that is limiting the amount of productivity, you can play with that nutrient and even act on large scales, such as this example in which iron salts were put into the oceans and the photosynthetic efficiency increased a lot. So, this is known as iron fertilization. Next, we had a look at nutrient cycles. So, nutrient is a substance that is used by an organism to survive, grow, and reproduce. You have macronutrients and micronutrients. Macronutrients are required in large amounts, micronutrients are required in smaller amounts. Essential nutrients are those nutrients that the organism cannot live without, and these cannot be substituted. Then we looked at the roles of some essential elements and the list of macro and micronutrients. Now, the plants need to have an access to all these nutrients, but then the earth does not have an infinite supply of these nutrients, because of which the nature has to recycle these nutrients in the form of biogeochemical cycles, which is a pathway in which by which a chemical substance moves through the biotic and abiotic compartments of the earth. So, this is a typical nutrient cycle. You will have a pool. From this pool, the producers or the plants are taking up nutrients. From they are also using the energy from the sun, and from the plants it goes to the herbivores, from there to the carnivores, from all of these it goes to the decomposers, and through these decomposers these nutrients again reach to the nutrient pool. So, this is a typical generalized nutrient cycle, and then we looked at specific cycles such as the nitrogen cycle, and we also looked at the industrial processes of nitrogen fixation. We looked at carbon cycle. Now, in the case of carbon cycle, all of these organisms are also directly putting in this carbon back into the atmosphere through the process of respiration. So, that is one difference that we have, and then in this case you have different pools. So, you not only have the pool in the atmosphere, but you also have a pool in the rocks, you also have a pool in the ocean waters, you also have a pool in the form of fossil fuels. Then water cycle, phosphorus cycle, sulfur cycle, and then we looked at the impact of humans, especially the Kagzera curve. So, if the plants are taken out, if you have a large scale deforestation or a large scale forest fire, the amount of water that will be flowing in your streams, it will go down, and then it will slowly start increasing and will take a very long period of time to reach back to its original levels. Then we looked at changes in pH and changes in the concentration of different nutrients in the water bodies. Next, fifth module was on population ecology. So, we began with population parameters and demographic techniques. So, population is defined as all the organisms of the same group or species which live in a particular geographical area and have the capability of interbreeding, such as the impala population, cheetah population, and so on. And this is the equation which is very important here. So, the population in the n plus 1th generation is equal to the population in the nth generation plus number of births plus animals that are coming in from outside minus the number of deaths minus the number of animals that are going out of this population to become a part of some other population. Then we looked at why these numbers are important and what are the kinds of demographic information that we are trying to get. So, we want to know the size of different populations, we want to know the density of different populations, and these densities can vary a lot. So, every different population may require a very different method for its measurement. Then we can have two kinds of densities, absolute density which is asking you the number of animals per unit area, and a relative density whether area X is more animals than area Y or vice versa. Then absolute population density can be measured in terms of total counts, such as the census of India, in which each and every individual is counted, or you can go for a sampling method in which you take a small sample of the whole population. So, this could include quadrates or capture-recapture method, and third one is the removal method, which we typically do not use in Indian context because we have a ban on hunting the animals. Then we looked at different kinds of quadrates. Then we looked at, because we are looking at sampling, we looked at the different processes that are involved in sampling. So, the objective of the sampling is to have a sample that is representative of the whole population. It is able to reproduce the important characteristics of the population as closely as possible, and at the same time it is not as intensive as measuring the whole of the population. So, we defined population, sampling units, and so on. So, all these definitions are important. Then the kinds of plots, you can have circular plots, rectangular plots, strip plots, or topographical units. Then we looked at different kinds of sampling, which is very important. So, we have simple random sampling such as a lottery, or a systematic sampling in which you measure every kth individual, or a stratified sampling in which you divide the whole population into strata, or you can have multi-stage sampling, or you can have probability proportional to size sampling. Then we looked at capture-recapture method. So, in this case you capture some animals from the population at time 1, you mark them with something. So, you can typically put in some tags, or you can color them with certain dyes, and then you release them. So, once they have mixed with the whole of the population, then you take another sample, and then in that sample you assess what are the number of animals that are having your marks, and then we can use this formula: number of marked animals in the sample divided by the number of animals caught in the sample is equal to the number of marked animals in the population divided by the total population size. So, we know the number of marked animals in the sample, the number of animals caught in the sample, and number of marked animals in the population. So, in that case we can get to the total population size. Of course, this includes a number of assumptions, and even the removal method. So, the removal method is not very applicable in the case of India, but then we also should know how this method works. So, we have these assumptions, and based on these assumptions we can say that if the population is changing at a particular rate, the number of animals that are being caught will depend on the number of animals that are actually present in that particular population, and we can integrate this equation to get to the curve that will give us the number of animals at every time point. Then if you want to measure the relative population density, you can use traps, fecal pellets, number of counts of vocalization and so on. The other things that we want are age pyramid, crude birth rate, crude death rate. So, all these definitions are important here. The general fertility rate, age specific fertility rate, total fertility rate, replacement level fertility, juvenile mortality, infant and life expectancy. And in the case of life expectancy, we divide it into physiological longevity, that is the life of an individual if it is free of any predation pressure and is living under optimum conditions. But then in the forest or in the jungle, all the animals are not able to live to their physiological longevity because they are not living in the most optimum conditions. So, the empirical average longevity of individuals of a population under given conditions is known as the ecological longevity. Then we want to note the immigration, emigration, net migration, natural increase, population growth and growth rates. Then we looked at the difference between precision and accuracy. So, if your measurements are close together, we say that they are precise. If your measurements are close to the actual value, then it is accurate. So, we want to have those measurements that are both precise as well as accurate. Now, if you have precise measurements but not accurate measurements, so you need to calibrate your method to overcome the bias. Next, we had to look at population growth and regulation. So, we looked at some numerical examples of how to measure the mean sizes, mean population sizes, densities and so on. We also looked at some methods for sampling of rainforest reptiles and small amphibians, and also we looked at some pan traps and their workings. Now, in the case of the mark recapture species, we were capturing the animals, we were marking them, but then there are a number of organisms that have natural body patterns that can be used in lieu of marking the individuals one by one. So, for example, every tiger has a different markings on its body, every zebra has a different marking on its body, or some animals that do not have such different markings such as the lion, you can make use of some other body part, such as you can make use of their vibrissae pattern, that is the pattern that is there in their moustache. Even some toads, frogs, spotted deer, they all have different markings on their body, and even your reptiles such as crocodiles and snakes, some of them may also be having different body patterns. So, we can make use of them in the monitoring of these population sizes. Then we had a look at the rate of population growth, and here we said that the population size at generation t will be at generation t plus 1 will be equal to population size in generation t multiplied by a growth rate of the population. So, in this case we can have a curve that is exponential. So, the population is increasing exponentially, but then in actuality your R naught or the growth rate is not constant. So, it varies with the population size, so that you get to the sigmoidal curve or the logistic curve. So, this is the logistic growth equation, dn by dt, that is the rate of change of population size per unit time, is a factor of the growth rate. It is a factor of the size of the population, because the more the larger your population size is, the more number of breeding females are there in your population. So, you will have more number of progeny, and but this factor is also modulated by this factor, which is a measure of the carrying capacity of your habitat. So, it is k minus n divided by k. So, we looked at the growth equation, this is how it looks. So, any population will start at very low numbers, you will have this lag phase. So, in the lag phase the population is not able to increase very much because you have very low number of breeding females. Then it will enter into this log phase in which the population is increasing very fast, and then it will come to this steady phase where the carrying capacity of the environment is now becoming a hurdle for the future growth of the population. So, the population size has reached to the carrying capacity, and then we did certain numericals. Then we looked at the Lotka-Volterra equations. In the case of Lotka-Volterra equations, this is telling us the prey and the predator population. So, the population of the prey is governed by its own population size, which is telling you the number of breeding females that are there in the population, and at the same time it is also governed by the predator population, because the more number of predators you will have, the more will be the mortality of the animals, because more number of animals will be eaten up for food. Similarly, the size of the predator population, it will increase based on the size of its own population, which will tell you the number of breeding females, plus it will also depend on the size of the prey population, which will tell you the amount of food that is available, or it will be an indication of the carrying capacity of the environment. So, if you put both of these together, you will get to a situation where both the prey population as well as the predator population, it undergoes a cyclical phenomenon, it goes through cyclical movements. And we can also make use of these Lotka-Volterra equations to look at the impacts of two different organisms on each other. So, say two different herbivores on each other, and we looked at this numerical example as well. Next, we looked at population studies and the equilibrium, in which case we talked about the problems of dynamics and the problems of statistics. The problems of dynamics are how does a population change with time, the problem of statics is how does the equilibrium condition and the average value, what does it depend on. So, in the case of dynamics, we have to ask these three questions: does the change occur in a particular time of the year, does the change occur in a particular stage in the life cycle of the organism, and what are the agents that operate at these times or in these stages. So, some common agents that can operate are extrinsic agents and intrinsic agents. Extrinsic agents are weather, predator, parasites, diseases, quantity and quality of food that is available, and shelter that is available. And intrinsic agents are physiological changes and the behavioral changes. In the case of statics, we say that the population size is not changing, so we do not have to consider the dynamic factors, but we can experimentally manipulate different habitat variables to look for the responsible factors that are responsible for this particular population size. And in this case, we looked at the locust problem, in which this species is found in two different forms. One is the solitary form, one is the gregarious form, and even though they look very different, they are members of the same species. And then we looked at how they change from one to another. So, the solitary phase moves via a transient phase into the gregarious phase, and the gregarious phase moves via another transient phase into the solitary phase. And when the population dynamics of these organisms were worked out, then we found that there is this particular formation of a plague, and this is the final outcome, that when you have a favorable weather, it increases the density of these insects, which then triggers gregarization, in which case the solitary phase converts into the gregarious phase. Once you have this gregarious phase, you start this vigorous cycle, because you have gregarious phase. So, this will give rise to more number of individuals, which will further increase density, it will further keep this gregarious phase on, and in this case you will have the insects that will form large size swarms and will start moving away. Now, this would continue for eternity were it not for the predators, were it not for the extrinsic factors that are also operating here. So, if the weather becomes bad, or if there is a large amount of predatory pressure, or if there are certain diseases that affect this particular insect, so in that case the population density will go down, and which will trigger a reverse phase transformation, in which the gregarious phase will convert into a solitary phase. And then these days we are also looking into the biochemical factors that are responsible for it. Next, in 6th module, we looked at community ecology. So, we began with community, nature and parameters. Community is an assemblage of populations of different living organisms that are living in a prescribed area or habitat, and the transition area where two communities meet and integrate is known as ecotone, which can be sharp or it can be diffused. Then we looked at community attributes. So, what are the things that are common between different communities? How do you define a community? So, it will have co-occurrence of species, these species are occurring together. There will be a recurrence of groups of the same species if you have two communities in two different areas which are having similar climate. So, you will have a recurrence of the species. And then a community is able to maintain itself, it is able to regulate itself through the process of homeostasis. Then there are community characteristics which tell you how two communities are different from each other, which are things like species diversity, growth form and structure, dominance, relative abundance and the trophic structure. So, they can vary between two different communities. Now, to describe a community, we have to describe the richness or the number of species that are there, the abundance of different species and the species dominance, which is the ranking as per the abundance in terms of number, basal area, etc. Then we defined the relative density, relative frequency and relative dominance. So, you can use this in especially in the case of tree species, and then you can get to the importance value index. So, a species that has a high relative density, which means that it has more number of individuals in that community. It has a high relative frequency, which means that the members of this species are found in all different parts of the community, plus it has a high relative dominance which have a high importance value index, and will say that this community is being predominated by this particular species. Now, if you look at the association between two species, we can make this contingency table, and if we have situations that in most of the quadrates you have two species either present together or absent together, then we will say that both of these species are positively correlated. But if you have situations in which one species is present, the other is absent, or the second one is present and the first one is absent, in that case we will say that these two species are having a negative association. So, this is an example of negative association. Then we looked at three community theories. We have the organismic theory, which was put forward by Clements, who said that the community behaves like a superorganism and it has properties that are more than the sum of its parts. On the other hand, you have the individualistic theory or the continuum theory, which says that the communities are just mere coincidences and there is no such organismic relationship. And then there is the third one, which is Hubble's neutral theory, which comes somewhere in between. It says that the species are functionally equivalent and the abundance of a population of a species changes by random births and deaths. Then we talked about guilds. So, guilds are a group of species which are utilizing resources in a similar way, such as a group of fruit eating birds in a rainforest, of forest floor dwelling birds and so on. Now, because species in a guild utilize the same resource, so the amount of competition between these species is much more than compared to any two random species. Then from guild we also define the niches. So, niche is a multi-dimensional description of a species resource needs, habitat requirements and environmental tolerances. So, if you have two species that are having the same niche, the level of competition is very high. And then in the case of these niches, we divide it into fundamental and realized niches. So, fundamental niches is a set of all the conditions that the species can potentially occupy, and we can determine it experimentally. But then in nature, when the species are competing with each other, then they are not able to occupy all the niches that are possible, and so the niches that are actually occupied go by the name of the realized niche or the post competitive niche. Now, we looked at the characteristics of niches and then the index of similarity. How many individuals are common between two communities will tell you the index of similarity. Next, we have association of a species with a community. So, you can have certain species that have a very high level of association, they are only found in a particular species in a particular community, or you can have lesser amount of association in the case of indifferent species which can be found in any particular community, or accidental species which are very rare species and they are either present as chance invaders from another community or as relics from a previous community. So, you can have different levels of association of a species with a community. Then we looked at community stability. So, stability is the ability of a community to recover following a disturbance, and it consists of two parts: resistance to changes and resilience to changes. Next, we looked at these community changes and ecological succession. So, from bare rocks you start getting some lichens. So, from bare rocks you have crustose lichens, then you have foliose lichens, then moss, then grass, then shrub, then forest, and all of and this process through which any area gets populated is ecological succession. So, ecological succession is the process of change in the species structure of an ecological community over time. And then we defined seer. A serial community or a seer is an intermediate stage found in the ecological succession in an ecosystem advancing towards its climax community. There are three kinds of seers: hydroseer, which is a community in water; xeroseer, which is a community in a dry area, which can be rocks or which can be sand; and haloseer, which is a community in a saline body such as a marsh. Next, we defined pioneer species. So, these are hardy species which establish themselves in a disrupted ecosystem and trigger the process of ecological succession. Now, typically these species have the ability to grow on bare rocks, nutrient poor soil, water. So, they do not have a very high nutritional requirement. They are pretty hardy species because they have to tolerate bare sun and maybe even very cold conditions, and they are also species that are able to release copious amounts of seeds or spores, because of which they are able to move into any new area. A climax species, on the other hand, is a biological community of plants, animals and fungi through which the process of ecological succession in the development of vegetation in an area over time has reached a steady state. So, this is the final level of succession. Now, climax community can be either a climatic climax, which is controlled by the climate, or it can be an edaphic climax, which is controlled by the soil conditions, or a catastrophic climax, which is controlled by some catastrophic events such as wildfires or floods, or it can be a disk climax, where it is controlled by some disturbance either because of men or because of domestic animals. Next, we looked at the characteristics of climax community. So, unlike the pioneer species, here you have vegetation that is extremely tolerant of this particular environmental condition. The species diversity is high. You have a well formed spatial structure, complex food chains which provide stability. There is equilibrium between gross production and respiration, uptake and release of nutrients. Species composition continues for a very long time. This again is an indication of the stability of the community, and it is a good indication of the climate and other conditions of the area, because here you have species that are very much tolerant and very much adapted to the particular conditions. Now, you can have three different kinds of succession. The primary succession is a succession that occurs in an area that was not previously occupied, such as a lava flow which has turned into a rock, and this rock was never occupied previously. A secondary succession is the successional dynamics that follows severe disturbance or removal of a pre-existing community, such as a forest fire. And a cyclical succession is periodic changes that arise from fluctuating species interactions or recurring events, such as flooding events if they are occurring regularly. Now, in the case of primary succession, we have this lithosphere primary succession that we have just seen, and the second one is the hydrosphere primary succession, in which you start with water, which has very low amount of nutrients. From water you move to phytoplankton stage, to submerged stage, to floating stage, to reed swamps, then sedge and meadows, then woodland, and then finally the climax stage. In the case of secondary succession, you can start with a forest. This forest has suffered from a forest fire, and then you have a forest that is incompletely destroyed, and then begins with the succession with the herbaceous stage, then the shrub stage, then the woodland stage, and the climax stage. And typically the secondary succession is much faster than the primary succession, because you already have well formed soil, you already have nutrients, you already have spores and seeds, probably some plants that can regenerate from their roots, and you also have a very high amount of fertility that can support a large number of organisms. Now, succession can also be divided into autogenic succession and allogenic succession. So, autogenic succession are brought by changes in the soil that are caused by the organisms that are already present there. So, auto is self, gen is formation. So, it is a succession that forms by itself. On the other hand, the allogenic succession is caused by some external forces, external environmental influences, and not by the vegetation that is already present there. Then we looked at these 7 stages of succession: nudation, migration, ascesis, aggregation, competition, reaction, and stabilization. Then we have these 3 theories of climax. You can have a monoclimax theory, where you have only one particular climax in one particular climatic area. You can have a polyclimax theory, in which you can have more than one kinds of vegetation climaxes, which are controlled by soil moisture, nutrients, topography, etc. Or you can have a climax pattern theory, in which you do not have one particular climax, but you have a range of climaxes which vary near the climatic climax. Next, we looked at community organization. So, there are two kinds of dominance for species. You can have a keystone species which is playing a much larger role in the ecosystem as compared to its numerical abundance, or you can have numerically abundant species which are determining the nature of the community, and so we say that they are the dominant species in that particular community. Next, we define the community dominance index, which is the percentage of abundance that is contributed by the two most abundant species. Now, if you have more number of species, typically the dominance reduces, but then this relationship is not a one-to-one relationship. If you have a community that has more amount of dominance, so it is possible that you will have certain species that are much more efficient in producing food, and in that case the productivity increases. Now, dominance changes with the surroundings. So, if you have nutrients that are put into a lake, that might result in an algal bloom. So, before this algal bloom you had a certain level of dominance, and because during this algal bloom a certain species has become predominant, so the dominance status of the community changes. So, typically you will also see that the species diversity is going down, then it increases again, and in a number of situations, if this perturbation is not very large, the community is able to bring itself back to the original position, which brings us to the concept of stability, which is defined as the ability of a community to defy change or to rebound from change. So, we have three different kinds of stability. You can have global stability, or you can have local stability, or you can have a situation where you have neither local nor global stability. So, in this case, if you have a community like this and you are giving it any amount of disturbance, if it is very small disturbance, then probably this community will be locally stable, but if it is any greater, this community will become unstable. Next, the question is, does diversity increase stability? We typically say that diversity increases stability, but there are also some counter arguments. So, this is still a matter of some debate. And how does a system respond to instability? You have resistance or inertia, which is the ability of the living system to resist the external fluctuations, or you can have resilience, which is the tendency of a system to retain its functional and organizational structure after perturbance of a disturbance. So, in the case of resistance, the community is not changing. In the case of resilience, the community has changed, but it is able to bring itself back to the original position. Now, resilience can be measured in terms of elasticity and amplitude. Elasticity is the speed with which the system returns to its original state, and amplitude is the maximum amount of disturbance that the community can tolerate. So, it is a measure of how far a system can be moved from the previous state and still return back to the original conditions. Then we had a case study of sewage in Lake Washington, and even after you had a large amount of disturbance, the community was again able to bring itself back to the normal conditions, which tells us that if you are within the limits, then the detrimental changes can be stopped and reversed. Now, in the 7th module, we looked at distribution and abundance of organisms. So, we began with biogeography, which is a study of the geographical distribution of life on earth and the reasons for the patterns one observes on different continents, islands and oceans. So, it asks the question, what is found where and why is certain thing found in a certain location and not in some other location. Then we looked at range. Range or distribution of a species is the geographical area within which the species can be found. Then we looked at major Indian habitats, alpine, meadows, forests. So, forests are also of different kinds, alpine, dry deciduous, moist deciduous, mixed forest, scrub forest and so on, sand dunes. Then we have estuaries, then run of Kutch gives a very different kind of habitat, then we have lagoons, we have floodplains, then we have shola forest, equatorial forest, mangroves, oceans and seas, and all of these are very different habitats from one another, and because of which we have very different kinds of species that are found in all of these different areas. Now, we looked at this example in which if you ascend in altitude, the temperature reduces, and so you can have those species which are typically not found in that particular latitude. So, if you go very high, then probably you will find even species that are more alpine even when you are close to the equator. Then if we look at the distribution of any organism such as snow leopard or the coral reefs, we are in the case of biogeography, we are first noting down where these organisms are found. Then we ask the question, what is there in this location that is making this organism come to this location or thrive at this location, and what is there that is not present at the other location where this organism is not found, because of which this organism is not found in that particular area. So, which brings us to the question of why things are where they are. So, we looked at these examples, and this is something that we have been looking for a very long period of time, and we define the factors that govern biogeography into push factors and pull factors. Now, pull factors are conditions that attract organisms to an area, good amount of food that is available, a good climate that is available to the organisms, and push factors are those conditions that drive away organisms from an area, such as scarcity of food or inhospitable climate. So, even in a small mountain we can see that there are certain areas where you have plants and there are certain areas where you do not have plants. So, these things can be right next to each other. Then we looked at the other factors. So, apart from push and pull factors, you have the factors of dispersal. It is possible that the organism has not yet reached that area, or you can have anthropogenic factors or man-made factors, in which case in this area plants should be there, but then because of the influence of man, this area was all cleared off the vegetation. So, you can have the anthropogenic factors. You also have the habitat selection factors, which refers to the hierarchical process of behavioral responses that may result in the disproportionate use of certain habitats to influence the survival and fitness of individuals. And habitat selection has both innate components, which are inborn components, and some learnt components. So, this was the example in which if you have wild caught adults of chipping sparrow, they spend majority of their time on pine and less amount of time on oak. But then if you rear them in the laboratory and you do not give them any exposure, they are able to repeat the same pattern. So, they will prefer the pine, but if you rear them in the laboratory together with the oak leaves, so in that case they will start preferring the oak. So, in this case this one is telling you that the birds have learnt to prefer oak. In this case we are seeing that there in the absence of learning there are certain inborn preferences that the birds have. So, the habitat selection has both innate and learnt components. Then we make use of transplant experiments to identify the potential range of an organism. So, if this organism is found in this green area, you can move an organism from one point in this green area to another point as a control experiment, or you can move it to other areas where it is not found. Now, if it is not found, there could be areas where it survives, there could be some areas where it does not survive, and based on its survival or its death, we can make some outcomes. So, if this transplant is successful, then probably the organism is not found there because this area is currently inaccessible, or it has not been able to disperse because of lack of time or because of habitat preference. But if the transplant is unsuccessful, then it could be limited by some other species, or it could be limited because of some physical and chemical factors. Then we look at factors. Now, next we move on to the Liebig's law of the minimum, which says that the rate of any biological process is limited by that factor in least amount relative to requirement, so that there is a single limiting factor. So, if you have n number of requirements, the one that you have the least amount of, that is going to be the limiting step. The second one is the Shilford's law of tolerance, which says that the geographical distribution of a species will be controlled by that environmental factor for which the organism has the lowest range of tolerance or the narrowest range of tolerance. Now, it is important to know these these different factors, because we are these days observing changes in the range. So, if you have global warming, those species that could not live in the colder areas are now able to increase in their ranges. So, we are seeing that the median altitude for malaria is increasing with temperature. Now, one other factor that can regulate whether a species is found or not found is migration, which is defined as a regular and seasonal movement of animals, often along fixed routes. Now, the reasons are, you can have animals that are looking out for better resources such as food or breeding sites, or there are animals that are moving away because their original habitats are having a harsh climate. So, they are shifting to a more amiable climate. We looked at the example of demoiselle cranes. Now, moving back to dispersion, we have three different modes of dispersion. You can have diffusion, which is a gradual movement over several generations, or you can have a jump dispersal, in which you have a quick movement over large distances, often across unsuitable terrains, such as the movement of zebra mussel through the ballast water. Or you can have secular dispersion, which is diffusion in an evolutionary time, so that the final migrants are very divergent from the original population. Then we also had a look at allelopathy, in which there are certain organisms that are secreting certain chemicals that are inhibiting the growth of certain other organisms. So, this is the distribution flow chart. Next, we had a look at some push and pull factors in more detail, in which case we looked at the distribution of sea urchins and algae. So, in this case we are saying that wherever you have sea urchins, you do not have algae, and where you have algae, you do not have the sea urchins. So, this led to a study of whether predators can restrict the ranges of the prey organisms. So, a certain area was cleared of the sea urchins, and if you remove the sea urchins, the algae are able to come back to that area, which shows that there is nothing other than sea urchins that was wanting in that area. Then, if you have a situation where a predator is governing the distribution and abundance of prey, so in that case you will have 4 conditions that will have to be fulfilled. The organism does not survive when transplanted to a site where it does not normally occur, unless it is protected from predators by cages. Second, there is an inverse correlation between the distribution of organisms and the suspected predator, or alternatively, in the places where it occurs, the organism is inaccessible to the predator. Third, you should be able to see that either in the field or in the laboratory, that the suspected predator is inflicting a lethal damage on the prey. And fourth, there should be a direct evidence that the suspected predator is responsible for the destruction of the prey in the transplantation experiments. So, if all these 4 criteria are met, then we can say that the predator is governing the distribution and abundance of the prey. On the other hand, you can have this counter example, in which case the prey is governing the distribution and abundance of the predator, which we saw in the case of Drosophila pachya, which is only found in those areas where you have a particular species of cactus. Now, does it have an implication on biological control of pest and invasive species? Yes, because if you have a predator that is extremely exclusive to the prey species, you can make use of that predator as a biological control. So, we looked at this example of controlling pine rust by eliminating the gooseberries, because the fungus that was affecting the pines also was obligately dependent on the gooseberries. So, it spent some part of its life cycle on the pine and some part of its life cycle on the gooseberries. So, if you remove the gooseberries, you will eliminate the pine rust. Then we looked at this other method of governing distribution, which is interspecific competition, in which you have a particular species of bird that is able to form colonies in this area, but then it gets displaced by another species because of interspecific competition, and this can also regulate the distribution and abundance of a particular species in a particular area, because once this species is displaced, now you do not have any individuals that are left in this particular area. Next, we looked at experimental demonstration of allelopathy. So, in this case, if you have the apple seedlings and you give it water, these apple seedlings are able to survive, but then if you pour water through a grass bed and then put this water onto the apple seedlings, they start dying off. but then if you put water only through a soil bed without the grasses, these apple seedlings are able to survive. So, this is telling you that there is something that is there in grass which is leading to the death of these apple seedlings here, because there is nothing in the soil that is leading to the death of the apple seedlings. So, this is how you can look at the impacts of allelopathy, or you can experimentally play with the impacts of the allelopathy. Now, in the case of allelopathy, it is not only that you have an impact on members of other species, but you can also have an impact on the members of your own species, such as this peach replant problem. If you have peach that is following an apple plantation, you have a good yield of fruits, whereas if you have a peach that is following a peach, you will have a bad yield of fruits. So, there is essentially something that is released by the peach plants that is inhibiting the other peach plants. And we looked at another example, in which case Crivella robusta kills its own seedlings. Now, there was one other variation that we looked at. Whenever we are talking about the range of tolerances, those range of tolerances can be modified by acclimation of individuals. So, if you have individuals that have been living in higher temperatures, they will probably be able to tolerate much higher temperatures as compared to other individuals of the same species that were living in lower temperatures. Then we also looked at one other variation, that several factors can be simultaneously governing the presence or absence of a particular species in a particular area. So, especially in the case of these tree lines. So, tree line is the line above which there is no growth of trees. So, that can be because of lack of soil, drought, desiccation, lack of snow, excessive snow, short growing season, rapid heat loss at night, excessive soil temperatures in the day, or mechanical aspect of high winds. Now, which of these factors is playing a role, or which combination of factors is playing a role, that needs to be looked at experimentally. And then we also saw that some amount of these variations are also stored genetically. So, for instance, if you take plants from different areas and these plants belong to the same species, and you are bringing these plants into the same area, they will start showing different features. So, whenever we are talking about these distributions, ranges, tolerances, so that is also modulated by the areas in which these organisms have been growing. So, that is all for today. Thank you for your attention. Jai Hind.